I will now proceed with the lecture. Now we come to a new topic which is known as weighted gene co-expression network analysis. It actually also has another name, which is weighted correlation network analysis. But the acronym is WGCNA. And as many of you know, WGCNA is also an R software package. And originally, we developed these tools for gene expression data, and that's where the name came from, weighted gene co-expression network analysis. But of course, lately we have applied it to many other omics data sets, notably DNA methylation data, microRNA data. We also applied it to functional MRI data, peptide count data, many other data modalities. And therefore, we decided to change the name to weighted correlation network analysis, because the essence is really to transform correlation coefficients between variables. And so, as I said, we will make all of this material available on multiple web pages. Here's one link, which is not the official course link, but it's just my own link, you know. And so today I will talk about various topics in WGCNA. One is how we construct such a weighted network, why we use soft thresholding, how we detect network modules, how we incorporate clinical traits, how we screen for important variables, and so on. So just to remind you of gene expression data and the standard differential expression analysis. So here we see a heat map plot where the rows correspond to genes and the columns could be different samples. So for example, controls versus an experimental perturbation. Red indicates, of course, overexpression. And now, if you produce a p-value or a fold change for every single gene, then you will get tens of thousands of significant results. But you really fail to see the forest for the trees, which is there are really only two patterns, you know, either genes that are up-regulated in the experimental condition and those that are down-regulated. And the idea of these systems biologic methods, for example, WGCNA, is to really pull out these patterns or clusters, really, and to focus on those. And now clusters have a special name in the context of network analysis, which is modules. People often call them modules. Modules is one of these overused words. You know, it has many different meanings. But here, in the context of WGCNA, a module is often a cluster, for example, found using hierarchical clustering. In any event, the philosophy of WGCNA is to understand really the systems aspect of it. And to use an old cliche, when you want to understand the functioning of an engine, you don't begin by enumerating individual nuts and bolts. You look at components. And so similarly, in the context of WGCNA, we don't focus on individual genes, but we rather focus on larger structures, which are these modules. And this has one major advantage, which is a statistical advantage, namely that we alleviate the multiple testing problem that plagues many genomic analyses. You know. So instead of focusing on 50,000 transcripts or 500,000 DNA methylation probes, we may focus on 100 modules or so. And the other thing is, we of course use here network terminology because it's very intuitive to biologists. Actually, these network methods are very intuitive to everyone because as human beings, we function in social networks. We have a tremendous intuition of what is a clique of people, a group of friends, which corresponds to a module, Or we know what is a hub. What is a hub? Well, it's a very popular person that has a lot of connections to many others. And so, although many of the techniques relate to standard data mining techniques that you may learn in computer science or in statistics, they are so intuitive that even people with minimal training manage to understand what's going on. And some of the most expert users of WGCNA never took any statistics class. They just become very good at using the software. The critical part of WGCNA is really the biologic interpretation. That's the bottleneck. So what is WGCNA? It typically proceeds along these steps. So we start out constructing a network among genes, for example. And the rationale here is to make use of the interaction patterns between genes. Most genes don't function in isolation. They really form pathways, or they are organized in certain ways. And so it's beneficial to exploit that. In step number 2, we identify gene modules. And the rationale here is, of course, that this is a module-based analysis. We focus here on these higher level structures. Now, in step 3, we relate the modules to external information. For example, clinical data such as disease status or plasma levels of certain biomarkers. But also, we could relate them to genetic markers or to gene ontology information. And the rationale here is to find biologically interesting modules. As you saw from the previous talk, when we identify modules, we use hierarchical clustering, and typically all we know is, well, this is branch 1, this is branch 2, and so on. And so to understand what's the biological meaning of these clusters, we need to carry out this step 3. In step number 4, we study the module preservation across different data sets. And why is that? Well, we do it because we typically want to check the robustness of our modules. If we have one lung cancer data set, we want to know which of the modules can be found in the second lung cancer data set. And just to convince the reviewers of our articles that what we have found is not some artifact. But also, it can be very exciting to study module preservation across different conditions. For example, we can ask which human brain modules can also be found in chimpanzee brains. So in any event, after these 4 steps, we hopefully found robustly defined, biologically interesting modules, and only then do we care to find out, well, what are the key drivers of these interesting modules? So are these modules enriched with certain do they contain certain transcription factors? Or we could ask, are they hub genes inside these modules? Or later in the course, you will learn techniques for causal testing analysis. You can ask which genes cause changes in these module genes, and so on. And the rationale here is to carry out experimental validation experiments or possibly develop therapeutic targets or biomarkers. So these weighted correlation networks are valuable for a biologically meaningful reduction of high-dimensional data. I mentioned gene expression data, not only microarray data, but many people have used these methods with great success on RNA-seq data. And also, they have proven their worth with gene methylation data. It turns out DNA methylation data show even more distinct module structures. CPGs are even more highly correlated than mRNAs and transcripts. In any event, the second major reason for using WGCNA is to integrate what we call multi-scale data. So it could be expression data from different tissues, brain tissue, blood tissue, liver tissue. or to relate gene expression data to genetic markers of interest, SNPs, and of course complex phenotypes. So many people have used WGCNA. It started out by people who study metabolic syndrome and mouse genetics, and of course Dan Geschwind and many other neuroscientists have applied these tools to neuroscientific applications. And during this course there will be several talks from neuroscientists. So how do we construct a weighted gene co-expression network? Let's start out even with what is a network? Well, for us, a network is specified by a matrix, and this matrix is called adjacency matrix. And so this matrix is typically a symmetric matrix because we deal with undirected networks, and the entries of that matrix take on values between 0 and 1. And for an unweighted network, the entries are 1 or 0, indicating whether two genes are connected or not connected. Whereas for a weighted network, the entries are real numbers. They measure the strength of the connection. So, for example, when we start out with multiple microarray data, but it could be again RNA-seq data, but multiple samples, then let's say we focus on two genes, and we plot their gene expression levels here on the y-axis versus the different samples. Let's say we have 50 samples. And let's say we have these two genes colored in green, and in the first person they take a low value, in the next person they take a higher value, in the next person they take a low value again, and so they go up and down together across these 50 people. Then we say that these two genes are co-expressed across the samples. And therefore we would like to connect them, because although we don't know why they are co-expressed, but they are co-expressed, and so arguably there is some sort of a connection between them. And now, how do we measure co-expression? That's a critical question. And in the course you will be exposed to many ways of measuring co-expression. But on the simplest level, we just use a correlation coefficient. For example, the Pearson correlation or the Spearman correlation. Now, as you may remember, the correlation is a number between -1 and 1. And why? Because it can distinguish between positive and negative relations. And therefore, a correlation is not a network adjacency. Remember, an adjacency is a number between 0 and 1. So we need to transform this correlation. And so in a second I will show you how to transform it, but after the transformation, then you have a network adjacency. So in this weighted network analysis, we take a holistic view depicted on the left, meaning everything is connected. The only thing that differs is how strongly things are connected. So for example, here are two genes that have a thin connection, whereas here are two genes that are strongly connected. Whereas in an unweighted network, there's just an edge. Two things are connected, yes or no, Now, let me describe how we construct a weighted correlation network. So when we have a correlation coefficient, which is a number between -1 and 1, we have two options. We can define an unsigned network or a signed network. An unsigned network treats positive correlations the same way as negative correlations. And you can see that here, because we take the absolute value of the correlation coefficient in an unsigned network. Meaning, if you have a correlation of plus 0.8 or minus 0.8, you get the same adjacency. Now, what you notice is there's this little power beta. And what is that? The purpose of this beta is that it is a threshold. The higher beta, the more emphasis is placed on high correlations. Now, in a signed network, we use a slightly more complicated transformation of the correlation coefficient. And the purpose here is to transform the correlation coefficient so that it lies between 0 and 1. Now, in general, I should have mentioned it, if you have a number that lies between 0 and 1 and you raise it to a power, then the result still lies between 0 and 1, you know. And so again, here we take a power beta, which serves as a threshold. And if you don't want to think much about it, then you just take these default values for the power. So a power of 6 for an unsigned network and a power of 12 for a signed network. The rule is when you go from an unsigned network to a signed network, you have to double the power. You know, that's the rule of thumb. And I will mention already that we do prefer signed networks. Why? Because we typically end up with modules that are biologically more meaningful. Yes, question? Yeah.
Just wonder, you know, when your correlation, the correlation between xi and j equals -1, you have alpha ij of 0, right?
Yes.
And if it's the correlation is 0, is alpha
yes.
ij still very close to 0?
Yes.
Right. I mean,
yes.
how do you distinguish?
Yeah, we don't. So just to repeat the question, because we're actually being filmed, you know. So the question is, when you use a signed network and you have a correlation of -1, then you can calculate that the adjacency is 0, you know, which is good, because when two genes are negatively related, we don't want that they end up in the same module in a signed network. But also, when you plug in a value of 0 for the correlation, which again, of course, indicates the genes are not related, and you calculate this, and you raise it to a power of 12, you again end up with an adjacency that is very close to 0. And so, again, these two genes would not end up in the same module because, you know, they have a low adjacency. And again, that's desirable, because if two genes are not correlated with each other, they shouldn't be part of a module, you know.
But the non-correlated
yes.
should be very different from negatively
yes.
correlated.
Yes. So if you want that two genes that have a strong negative correlation with each other end up in the same module, if you feel they should be connected, you better use an unsigned network. You know, it's really a decision the data analyst has to address at the outset. You know, and it's a biological question. It's not a statistical question. So later I will show you data that show that often modules correspond to cell types, you know, and then we often in this case, to find these cell type modules, we typically want a signed network, you know. Why? Because we want genes that are overexpressed in one particular cell type, you know. That would be a nice module. Having said this, imagine you have a single cell data set, meaning RNA-seq data from single cells, and hopefully later I show you such an example. Then in this case, you may want to find signaling pathways between genes in the context of a single cell. So when you have signaling pathways, it's often interesting to find genes that down-regulate others. And if you want to do that, then probably it's a far better exercise to use an unsigned network, you know, because then modules may correspond to pathways comprised both of positive and negative regulators, you know. So it's really a question. But I tell you this in my applications, which are typically from very heterogeneous tissues and so on, I find that in 90% of cases, signed networks are superior. And so I urge you, if you, to use those. But the good news is these analyses take, maybe 10 minutes, you know, so you can try both. There's one argument in our R function, which is called network type. And you just say network type equal to unsigned or network type equal to signed. And you, as a data analyst, decide what works better for you, you know. Let me proceed, and here I show you scatter plots where the y-axis is the adjacency, meaning the connection strength between a pair of genes, and the x-axis shows you the underlying correlation coefficient, which of course is a number between -1 and 1. And these different curves correspond to different choices of the power beta that I mentioned. Remember this power beta. And let's say we have a power of 1, then we get this straight line relationship, and it is unattractive. Why is that? Because notice if the correlation is 0, then the adjacency is still 0.5, meaning they are still connected, and that feels wrong. Things that are not correlated shouldn't be connected. So therefore, we really advocate that you pick a higher power, and you notice this effect. If you have a power, let's say 8, then yes, all correlations that are negative essentially end up with an adjacency of 0, which is desirable. Only positive or high positive correlations end up result in a high adjacency. Let's contrast these curves with another approach, which is to simply choose a threshold value of, let's say, a threshold of 0.8. So you have let's say a correlation coefficient, and you say if the correlation is bigger than 0.8, I connect these two genes, meaning adjacency 1, and if the correlation is less than 0.8, then you don't connect these, meaning adjacency 0. So what you see is that this hard threshold of 0.8 leads to a discontinuity. There's a jump, and that is unattractive. Why? Because it feels arbitrary. In certain ways, you delete information. Because imagine you have two genes that have a correlation of 0.79, then you say they are not connected. And if their correlation is 0.81, then you say they are connected, And given that the data are so noisy, that feels too harsh a split, And therefore, we prefer to map the correlation coefficient to the adjacency in a continuous fashion, right? This continuous curve. And this continuous curve is referred to as a soft thresholding approach. And so these weighted networks result from soft thresholding a correlation matrix. So here I talked about a signed network. Now for an unsigned network, you get curves that look like U's. You get a U-shaped curve. Why? Because both positive correlations and high negative correlations end up with a high adjacency. So you connect these two genes. Are there any questions at this point? Good. So we already discussed this question. Should network construction account for the sign of the co-expression relationship? And here's an article I cite, but I could cite many others, where we found that using signed networks was really critical, to find biologically exciting modules, And therefore, we prefer it. So another question is, why do we construct a co-expression network based on the correlation coefficient? As I mentioned, later in the course, we describe many other methods. But there are good reasons for focusing on correlations. The first is, it's of course very intuitive. Even people with a minimal training in statistics know what it is. Now, the Pearson correlation coefficient measures the extent of linear relationships. And so if you have a relatively small sample size, let's say 20 samples, then focusing on measuring linear relationships avoids the pitfalls of overfitting. Of course, also these correlation networks have performed exceedingly well in practice. They're computationally fast and they lead to reproducible research. I should mention one thing at this point. So many people criticize correlations because they say, well, this measuring linear relationships seems a bit unbiological, because clearly we know they're nonlinear relationships. But let me mention, of course, there's another correlation coefficient called the Spearman correlation, and that measures the extent of monotonic relationships. So if you care to capture monotonic relationships, just use that. Having said this, very few people use the Spearman correlation in practice, and there's a reason for it, because somehow it seems to delete signal. It's overly robust, because the Spearman correlation replaces each expression value by the rank of that expression value. You lose something for that. And therefore, what we would really like to have is a measure that is a bit like the Pearson correlation in the sense that it maintains a lot of the information, but a bit more robust than the Pearson correlation and captures more monotonic relationships and is less susceptible to outliers. Now, fortunately, we have such a measure. Actually, it's been known since the 1970s. There is something called the bi-weight mid-correlation. It's described in many textbooks. For example, here's one from 1977. And so it is a robust alternative to the Pearson correlation. And one idea is, just remember, in the Pearson correlation, you somehow form the covariance, and the covariance is based on subtracting the mean from each observation. So in the bi-weight mid-correlation, for example, we subtract the median value, and also we weigh observations in a fashion so that observations whose values are close to the median receive larger weights. And in the WGCNA package, Peter Langfelder implemented a fast method for calculating the bi-weight mid-correlation. And also, most of our R functions that build correlation networks have an option where you specify the correlation type. It's a simple argument. You can say core type equal bi-weight, and you get that. Or core type equals Spearman, or core type equal Pearson. So it's you can play around and decide which approach is best for your data. But I tell you already that I'm of course often lazy about that, and I just go with the bi-weight mid-correlation or the Pearson correlation. I never even explore the Spearman correlation because I tried it many years ago and I didn't like it, and so we are all very busy people, So, so I do want to mention one thing. There has been quite a lot of interest of defining co-expression measures. Later in the course, I talk about a measure called mutual information that is familiar to many computer scientists. And then there are many alternative statistical dependence measures. And fortunately, I convinced Lin Song to carry out a completely unbiased analysis, an empirical evaluation on 8 different applications, ranging from brain cancer to mouse genetics. And we judge different methods with respect to the biologically meaning, or more precisely, the Go enrichment of the resulting modules. And to tell you already, it turned out that her studies indicate without a doubt that using the bi-weight mid-correlation coupled with the so-called topological overlap measure worked best for finding modules that were enriched with significant Go terms. And I'll show you these results later in much greater detail, But I wanted to mention it at this point because I hope I can pique your interest to learn what is this topological overlap measure. I'll explain it to you in a second. So, although these methods that I present here are quite simple, don't think they are unsophisticated or that they're more. Let me rephrase it. They're statistically more sophisticated measures, but we have empirical evidence that these more sophisticated approaches may overfit your data. Especially if your sample size is limited. So why do we use soft thresholding as opposed to hard thresholding? Well, as I mentioned, one reason is that this preserves the continuous information of the co-expression information. Co-expression is measured using a continuous measure, so why threshold it? Also, we have empirical evidence that shows that the results tend to be more robust with regard to different threshold choices. So, as I said, these default choices for the power work really well. If I choose a power of 12 for a signed network, things really don't change much if I use a power of 8 or a power of 15, you know. And so that's a major advantage. You don't have to think much about it. In contrast, if you use a hard threshold of 0.8 or 0.5, you really get very different modules. Now, the hard thresholding, though, has its own distinct advantages. In particular, there are many graph theoretic algorithms from the computer science community that you could apply, and also you can visualize these networks, you know. So we also visualize these unweighted networks. For example, you can do that with Cytoscape and Visant, and later today and tomorrow you will learn about Cytoscape, you know, so you learn that. So continuing, what are the advantages of using soft thresholding with the power function, meaning we raise the correlation to a power? As I said, number one, robustness. But there's a second reason, which you will appreciate in the afternoon, which is that we can calibrate different networks. For example, imagine you have 8 different lung cancer data sets that you downloaded from GEO, for example, or from different citations. And some of these lung cancer data were measured on the Affymetrix array, others measured on the Agilent arrays, others using RNA-seq. Some of the data come from Caucasians, others come from Japanese and different ethnic groups. So what you would like to do is so for each of the 8 data sets, you get a separate network, 8 different networks. And you may say, I want to combine these data into an overall network, which you call the lung cancer network. Now, a critical step to accomplish that is to put these data on the same footing. You need to make them comparable, which I call calibrate these networks. You could call it normalize these networks, you know. And in order to calibrate then these network adjacencies, it really is very valuable when the adjacencies are real numbers between 0 and 1, meaning a weighted network. Why? Because you can transform them, for example, using a power transformation. And in contrast, if you have an unweighted network where all entries are 0 and 1, you really have a hard time calibrating these methods, and you need to then use more sophisticated weighting schemes. Then the third reason why we like these weighted correlation networks is that they give rise to powerful module preservation statistics. So in a separate lecture, I will teach in great details how we measure whether one module that was found in the first data set, whether this module is also preserved in a second data set. And I will show you that weighted networks give rise to powerful statistics. The fourth reason is a mathematical reason. So one can show that when you use this power transformation, that you can derive beautiful theorems if you care to do so, you know. There are actually mathematicians who like correlation networks because they allow one to prove theorems that are very hard to prove in other networks, you know. And I think on Friday I briefly touch on that, you know, but I don't want to traumatize you with it, because if I give you here too much math, you will change your flight ticket, you know. So in any event, let me come to how do we choose the power, the threshold, or in general, how do we choose the threshold for a correlation matrix? And now when you have a hard threshold for a correlation coefficient, you can actually calculate a corresponding p-value or a q-value. And you use a statistical criterion. However, and that's good. However, for weighted networks, that approach doesn't work because there is no hard threshold. And therefore, we need a different criterion. And one idea then is to make use of properties of the connectivity distribution, and I'll describe that now in greater detail. What do we mean by connectivity? So the connectivity is really the row sum of the adjacency matrix. So here's a formula if you know how to read that. But what is it intuitively? If we're in a social network, my connectivity is the number of friends that I have. And so for an unweighted network, it's just the number of direct neighbors. But for a weighted network, it's the sum of connection strength. And what you can do, so you have this by the way, connectivity is also known as the degree when you deal with an unweighted network. So some people who study graph theory are more familiar with that term. But in any event, so we have this connectivity for every gene, we measure its connections. And now what we can do is we can form a histogram. So the x-axis shows the connectivity and the y-axis shows the frequency of the connectivity. And often when one plots this graph, this frequency plot, you see this highly skewed distribution. And to make a very, very rough statement, when you have this very highly skewed distribution, then we say the network is approximately scale-free. That's a very rough statement. Let me give you a more precise definition. Notice here I plot connectivity versus frequency of connectivity. What one typically does is one actually creates this plot on the log scale. So logarithm of the connectivity versus logarithm of the frequency. And you get a plot, and if you get a straight line relationship, then we would say the network satisfies scale-free topology. That's a more precise measure. Now, in statistics, if I ask you, how do you measure that two variables follow a straight line relationship, then hopefully you remember, well, the Pearson correlation. That's actually the optimal measure for measuring straight line relationships. And so that's really what we do. So we take the Pearson correlation between the log of the frequency versus the log of the connectivity, and this Pearson correlation square, the R square measure, that's the measure we use to measure to what extent scale-free topology is satisfied. So if this R square is high, for example, here it's 0.87, then we would say, well, it's at least satisfied approximately. It's a fairly good fit. Now, there are many more precise mathematical definitions of scale-free topology. As I mentioned, you can have, this is the definition log of the frequency forms a linear relationship with log of the connectivity. And there are alternative definitions. There's something called exponentially truncated scale-free topology and so on. And there's a vast literature on scale-free networks. As I mentioned, Albert Barabasi and his colleagues explored, going back a few slides. So Dr. Barabasi wrote several papers on properties of scale-free networks. But we use this scale-free topology property, this R square measure, to decide how to choose a threshold, and I'll get to that in a second. Let me mention already that there are many networks that do not satisfy scale-free topology. So for example, here I have a yeast co-expression network, and notice the dots don't form a straight line, they form a quadratic relationship, And so just be aware of it, that co-expression networks not always satisfy a scale-free fit. Sorry. Now, there have been many articles where people studied their favorite network, and then they said, well, isn't it interesting? I find the network satisfies scale-free topology. However, we noticed very early on that this is a statement that is highly dependent on the threshold you choose. So if you choose a low threshold, for example, a power of 1 for the soft threshold, your network doesn't satisfy scale-free topology. But if you choose a threshold of 20, then yes, it satisfies scale-free topology. And therefore, we said, well, instead of writing a paper, my liver expression network satisfies scale-free topology, we don't make this claim. We rather are honest about it and tell the reviewer we choose the power so that the network will satisfy scale-free topology approximately. Do you see? That's the idea of the scale-free topology criterion. And but why do we even want it? Going back to the frequency plot, let's think about what it means. So what does it tell me? It tells me that the vast majority of genes has low connectivity, which in many cases is a biologically reasonable statement because many genes don't do anything. They just do nothing in the background. However, there will be some genes that, of course, are highly related to hundreds, possibly thousands of other genes. And these highly connected hub genes can be found here. But in many applications, you would want that there are few such hub genes, not in all, but often that's what you want. And therefore, if you build a network, you can say, well, I have no reason to expect that there are many hub genes. I want few hub genes. It's biologically more plausible. And if you make this assumption, then you want to choose the threshold so that your network is approximately scale-free. That's the logic. We developed the scale-free topology criterion for choosing the parameter values of an adjacency function. And that is, consider only those powers beta so that you get an R-square index that is reasonably high, so that you have scale-free topology. So let me be much more concrete by focusing on this slide. So here I show on the y-axis the R-square index measuring scale-free topology, and on the x-axis I show different powers. And as I mentioned to you, when you choose a low power of 1 or 2, your network doesn't satisfy scale-free topology. But as you increase the powers to 6, 7, and 8, suddenly it seems to satisfy scale-free topology, And therefore, one could, when you have such a curve, you could say, well, okay, I just take a power of, for example, 7, but clearly tell the reviewer, I have scale-free topology because that's how I constructed the network. Now, in practice, we often see such a relationship where for different powers, initially R-square goes up, but then often it tapers off or it even goes down. And therefore, to be more precise, the scale-free topology criterion chooses the lowest number where you get a high value, where the saturation starts. That's how I use it. Now, the other thing to mention is that the mean connectivity, of course, decreases, right? As you increase the threshold, the number of connections diminishes, and so you get this plot where the mean connectivity is monotonically decreases for different powers. So going back here, so why do we use the scale-free topology criterion? Well, it's an empirical finding that in many co-expression networks based on expression data from a single tissue, for example, blood expression data or liver expression data, you find scale-free topology for reasonably small powers. 6, 7 for an unsigned network, or 12 for a signed network. And sometimes you really, but sometimes you don't. And I get probably one email a week where somebody shows me a scale-free plot and says, I don't get scale-free topology, you know. And then my response is always the same. What's your sample size, you know? Because sometimes people apply these methods to very small data sets, let's say 10 samples or less. And there's a problem with that. You would want, I want to say 20 arrays or 20 samples. But the other big reason is that they may have an outlier, you know. Let's say they have a tissue, brain tissue, and everything, it's this, let's say cortical samples. And the truth is I would want to see scale-free topology. Why? Because we always see scale-free topology for reasonably small powers. And if they don't see it, then my next question is, did you cluster the samples, meaning the cortical samples, to see whether there are outliers, you know? There could be data quality issues. There could be batch effects. So technical artifacts could lead to a correlation pattern among genes that doesn't allow you to find scale-free topology. But there is also a biologically good reason why scale-free topology couldn't be satisfied, which is, imagine you have two distinct tissues. For whatever reason, you analyze liver tissue and adipose tissue together. And in this case, if you do that and you form a co-expression network, you will end up with modules that are very large. There could be a module containing 10,000 genes. Why? Because this is the module of all genes that are overexpressed in liver tissue. And then there's another module of 10,000 genes, that's all the genes overexpressed in adipose tissue or liver, you know. And so when your data set, for whatever reason, contains a few very large modules comprised of tens of thousands of genes, then what does that mean? It means you have tens of thousands of hub genes, right? Because if a gene is a member of such a large module, it's highly connected to thousands of other genes, and therefore it's a hub. And so therefore you get thousands of hubs, and therefore you will never get scale-free topology, because as I mentioned, one implication of scale-free topology is that you have relatively few hubs, you know. So don't despair in this case, just go with the default choices of the power that we have, right? So for a signed network, just proceed with the power of 12, you know. Are there any questions at this point about, okay. So let me move to another topic, which is how do we measure interconnectedness in a network? Interconnectedness is a vague term, you know. Things are interconnected. What does it mean? Well, it really means connected, right? And well, I hope you know the number one answer. The adjacency measures connections, right? That the adjacency measures the connection strength. But we actually have a second measure of measuring connection strength, which is called topologic overlap matrix measure. And this is a little bit more complicated, but it has a very nice intuitive explanation. So let's think of a social network. Where the adjacency measure specifies whether two people like each other. High adjacency means warm relationship. Now, it could be that in one particular week you ask two people, do you like each other? And they say, oh no, I really dislike this person because they stood me up. We had an appointment and they feel they are better than me and didn't show up, you know. So it's a vague measure. It's too unstable. But if you ask the different question, which is, how many friends do you share in common with the other person? The shared friends, that's a very stable measure. Because if you share the same group of friends, you will meet the person again, and chances are after 2 weeks you make up and you like them again, you know. So looking at shared friends is even in social networks, possibly a more robust and even more meaningful measure of measuring interconnectedness. And so the same actually applies also to gene expression networks. As I mentioned to you earlier, we have a scientific paper where we carried out a comparison study, and we could show that if you measure shared neighbors of genes, you end up with modules that are often biologically more meaningful. And therefore, let me explain to you this topologic overlap matrix measure. So the topologic overlap measure is defined, if you want to be very rough about it, it's the matrix product of the adjacency matrix with itself. That's it. But you normalize that matrix. This is the matrix product of two genes I and J, but you normalize it in a way so that the result lies between 0 and 1. Okay? Now, so what we do is we divide by the minimum of the connectivity, and this formula incidentally assumes that the diagonal elements of the adjacency matrix are 0. Now, the topologic overlap measure that measures shared friendship is a similarity measure. And as Peter mentioned earlier, when we do clustering, we need a dissimilarity measure. Why? Because most implementations use a dissimilarity. And therefore, we turn this similarity into a dissimilarity by taking 1 minus it. Now, the topologic overlap measure also has a nice set theoretic interpretation. Oh, by the way, I forgot to mention there's a science article by Horvath et al. Where the authors introduced this topologic overlap measure in the context of protein-protein interaction networks, where it also works, of course, very well. But here we recognized that this measure also works well in the context of co-expression networks. But coming back to the interpretation of that measure. So let's assume we have here this network, and this is really an unweighted network. And what you can ask for this point 1, you can ask what are its direct neighbors, meaning which nodes have an edge with node 1. And this set of direct neighbors of the ith gene is denoted by that. So that's the set of nodes around gene I that are one step away, meaning one connection away. And so then the topologic overlap measure can be interpreted as the size of the intersection of the two neighborhoods of two genes, right? You have the neighborhoods of two genes, you overlay them, and you say how many genes are shared in common, and you ask the cardinality. And so in this sense, it has a nice set theoretic interpretation. And now the one thing to remember is that here we defined the neighborhood of the ith gene using one step, meaning the direct neighbor. However, you can, of course, generalize that and say, I define the neighborhood of the ith gene as any gene that is 2 steps away. So what we call 2-step neighborhoods. And so intersect that. And so you can generalize it. But I warn you that this approach is probably a bad idea. We never use it, although we tried, Why you over-merge things, Suddenly everything is highly interconnected. So in our applications, we only ever use the topologic overlap measure defined by using this one-step neighborhood approach. Any questions at this point? Okay. So how do we detect network modules? Remember, modules are quintessential for WGCNA. And as I mentioned, we often define them using hierarchical clustering. And so as Peter Langfelder reviewed earlier today, we get a cluster tree, and then the modules are labeled by integers and typically by size. Module number 1 is the largest module, module 2 is the second largest. And for the purposes of visualization, we typically assign a color. And so in the past, was this convention, and we still largely follow it. Turquoise is the largest module, blue is the second largest, brown the third largest, and so on. So as I mentioned initially, we know nothing about modules except these are branches, And earlier today, you heard a lecture on how one can cut branches from a hierarchical cluster tree. So this is a hierarchical cluster tree from an R software tutorial that I will present to you tomorrow. And these are mouse gene expression data from Jake Lucis's lab. And you see these branches in the tree. And as Peter reviewed, each branch is a module, it's a cluster. And so the challenge here is to cut the branches. And that is, there are several R functions. There's this static approach where you use a single height cutoff. And there are these dynamic approaches. Dynamic is here another word for adaptive, right? There's no time series. It's and what does it adapt to? It adapts to the shape of the branch and the size. There's certain parameters. And although there are several parameters, I want to emphasize that the default values work phenomenally well. Some people here in the audience are expert users of WGCNA, and they know how to tweak every parameter, and I applaud you for it. But the truth is, I barely touch it. I often don't touch it at all. And why is that? Because sometimes I have data sets that contain 500,000 variables, and I really can't even look at the tree, and so I just go with the default values. And the truth is, it works really well. So I do want to remind you that there are really two dynamic approaches. There's a method called the tree method, and one is called the hybrid method. The first method is called the tree method. Why? Because the input is just a hierarchical tree. Actually, any way for you to create a hierarchical tree would be good enough. You don't have to use WGCNA to construct a tree. Some people actually do phylogenetics and use these approaches. Any tree could serve as input of this dynamic branch cutting approach. That's the first version. There's a second version of the branch cutting, which is called the hybrid method. Now that hybrid method inputs two things. It inputs the hierarchical cluster tree, but it also inputs the distance measure you used, or the dissimilarity. So, and why is that? Because it uses that distance measure in a second clustering approach for which could be interpreted as partitioning around medoids. So, so the hybrid approach, as indicated by its name, is that in the hybrid indicates that it's a hybrid between two approaches. One is hierarchical clustering, and one is partitioning around medoids. The great advantage of hierarchical clustering, there are two major advantages of it. Number one advantage is hierarchical clustering is very good at dealing with what we call singleton clusters, or background noise, really. Right? If you have 500,000 variables, then many of them are really not part of a cluster. They're background. They do nothing. And so you need a clustering approach that doesn't force them into clusters. That's number one advantage. The second advantage is that you don't need to specify how many clusters are in your data set, right? If you have 500,000 variables, can you honestly say that you estimate there are now 20 clusters in the data, or do you want to say there are 100 clusters? You really don't have a good sense for the initial number of these clusters. And so hierarchical clustering really frees you up from specifying it. Why? Because there are a bunch of branches, and the algorithm cuts them, and so you don't need to specify it. Now, what's the advantage of partitioning around medoids clustering? The number one advantage is robustness. it's the authors, Kaufman and al., who introduced it initially. That was their number one selling point, robustness. And so by marrying these two approaches, the hybrid method inherits the advantages of both approaches, hierarchical clustering and PAM. Let me proceed to another topic, which is how do we visualize networks? There are several approaches, and one well-known one, it has several names. There's something called the connectivity plot. We call it topologic overlap matrix plot. But what is that? So in the topologic overlap matrix plot, we visualize the topologic overlap matrix, right? Why? Because it specifies pairwise connection strength. But notice that the rows and the columns of this TOM plot are sorted, and they are sorted according to the hierarchical clustering tree. And now, for a statistician, the only part that matters about this plot is the hierarchical cluster tree, right? Why? Because that was used to cut the branches. That gives us the modules. But for one reason or another, people like to see colorful graphs, and so therefore they often show also the underlying connectivity patterns. And when you do that, then the modules correspond to clusters, to squares along the diagonal, right? So here you see this turquoise module, this very big branch, and it roughly corresponds to this square, Similarly, the green module gives rise to this square, All right. That's the TOM plot. Yes? Yes?
Can you say something about the properties of the genes that is at the very end of the branch?
Yes.
From a statistical and then from a biological standpoint, what do we infer from a guy that's at the bottom versus at the top of the branch?
Yes, thank you. So the question is, when we have a branch such as the turquoise branch, then there are, of course, some genes that are located at the tip of the branch, and others are way up. What's the difference? So there are many answers. Let's start out with this. When you are located at the tip of the branch, we are really sure you are a member of that branch, right? It's very intuitive, because no matter where you cut, the tip is always in the cluster. So in certain way, the statistical answer is these are the genes that have high module membership. They are certain. Whereas the genes here at the very top of the branch have a weaker connection to the cluster or the module. And so the truth is we are not really sure. Are you part of that or not? Now, there's another way to characterize them, which is using this measure of what we call intramodular connectivity. So the genes in the tip of the branch, they are the hub genes. They're the intramodular hubs. They're highly connected to other genes. Why do we know it? Because genes that are highly connected to all the other genes in the module, those are the genes that we are sure of. They are part of the module. Whereas the genes at the top of the branch, they have much weaker connections. They could arguably be misplaced. And so later we will introduce a measure of intramodular connectivity, and in essence it measures how deep you are located in the branch. All right. Visualizing. So this is the topological overlap matrix plot. Let's come to another visualization tool which we will show to you later today, which is multidimensional scaling. This is a way of projecting a high-dimensional space, in certain ways, into a two-dimensional or three-dimensional space. And so, to be more precise, a multidimensional scaling takes as input the dissimilarity measure and translates it into Euclidean distances. And so here we visualize the same network, and each dot is, of course, a gene. And you notice the modules roughly correspond to the fingers of a hand. And now, when we have these fingers, then the genes that are in the tip of these fingers, these are again the intramodular hubs. These are the genes you're really sure of are part of the module. Whereas there are some genes colored in gray, and they're in the center, and these are the background genes. Remember, we take the color gray to denote genes that are not part of any module. And as I told you, often you have thousands of genes that are not part of any module. They in certain ways cannot decide in which finger they should go to. Later today and tomorrow we teach you how to create these two plots. But then also you can use alternative software, for example Cytoscape or Visant, to create a traditional view of a network. So traditional meaning unweighted networks, there's just an edge. And so again, you see these modules, and here modules are just little fur balls, of clusters of points that are highly connected. Now, all of these are nice ways of visualizing things, but of course a biologist would like to know, so what's known about these genes? What is special about the brown module genes? What's their meaning? And to do that, one can do gene ontology enrichment analysis, and we'll teach you some of these tools. I already mentioned their web applications, for example, David or Ease and WebGestalt. And then there's commercial packages, Ingenuity, and so on. And they allow us to say, well, this set of brown module genes is highly enriched with mitosis genes, for example. So you arrive at that type of an interpretation. There's also another view of a co-expression module, which is the heat map view. So what is that? So here, the rows correspond to different genes. And for example, blue indicates all the genes in the blue module. And the columns correspond to different subjects, for example, different brain cancer subjects. And what we see is, we see these vertical bands, black and red. And we see, of course, green bands. So red and black indicates high values, and green indicates low values. And so why do we see these bands? Well, it indicates that genes go up together in some people and they are down in other people, right? Meaning these genes are co-expressed. And so that's of course no surprise, because if you have a module, then yes, the genes by definition are co-expressed. These are co-expression modules, so you see these bands. And when you look at this heat map, that looks good to me, meaning that these modules are biologically meaningful. However, sometimes you see really crazy heat maps, which could result from outliers, artifacts, and so on. And later, or tomorrow, I show you our software tutorial where we see a heat map that looks really bad. And so it's always a very useful exercise to look at heat maps of modules to make sure that things are kosher. Now, as I mentioned, often these modules are comprised of hundreds of genes, and we want to summarize the expression profiles in a given module. And this has been solved in two ways. There's a mathematical answer and there's a network answer. So the mathematical answer is you use the optimal way of forming an average of these genes. And that can be done using principal component analysis, or equivalently something called singular value decomposition. But there's the other answer is when you have a module, well, you take the one gene that is most highly connected in that module, the ultimate hub gene. And fortunately, both of these answers, for all intents and purposes, turn out to be equivalent. But let me explain to you what we mean by module eigengene. It's actually a crucial concept that I explain now. So this is now a very good time to wake up in case you've been checking your email. All right. So what is the module eigengene intuitively? So here I show again a heat map plot of the genes so the rows are the genes and the columns are different subjects. And so the heat map, this particular individual had most of its genes overexpressed. All of these genes take a high value. Now, the module eigengene, which is depicted right underneath this plot, takes a positive value for this person. Whereas here are subjects that where these module genes take low values. And you notice that the module eigengene takes negative values, right? So notice the bars of this module eigengene, the bars indicate the level of this eigengene in these different subjects, and so it measures in certain ways the overall expression of the module genes. Do you get that? So the eigengene is not a real gene. Sometimes people ask me, so what is the eigengene an apoptosis gene? But that's not a meaningful question. The eigengene is a mathematical construct. It's a weighted average of all module genes. And therefore, in this case, it would take 50 values because there are 50 microarrays. All right. So for the experts, let me mention that the eigengene is defined using the singular value decomposition. And so what do we do? So first, a first critical step is to standardize or scale each gene in the module so that its mean value is 0 and the variance is 1. Why do we do it? Because we want that all genes in the module are put on the same footing, irrespective of their mean expression values. After we standardize these genes in that fashion, we carry out the so-called singular value decomposition, which is up to a minus sign, really equivalent to principal component analysis. And then the singular value decomposition not only leads to what is known as singular values, but also to singular vectors. And then the eigengene is defined as the first left singular vector, meaning it is that vector that explains the highest proportion of the variance. Right? The idea is all of these genes vary, and you want a summary that summarizes this variance in an optimal fashion. And that's what it is. So now, why do we like eigengenes? They have many uses. But number one use is they allow one to relate modules to each other. Let's say you have the brown module and the blue module now, and you want to relate them. How would you do it? Well, you represent each of these modules by their respective eigengene, and you just correlate these eigengenes. And you could say, wow, these two modules are highly related because their eigengenes have a correlation bigger than 0.8. And if this is so, you could say, I should have merged the modules. They're really indistinguishable. The second reason why we like eigengenes is because we relate module eigengenes to clinical traits. We can say, well, which module eigengene correlates with HDL levels, or which module eigengene shows evidence of being differentially expressed between autism cases and controls. And by focusing on module eigengenes as opposed to individual genes, we avoid the multiple comparison problem. So, instead of relating 50,000 transcripts to disease status, we may relate as 50 modules, or more precisely 50 module eigengenes to disease status. The third reason why we like module eigengenes is because they give rise to this measure of module membership. So when we have a gene, we can correlate it to the module eigengene, and that correlation measures in certain ways how close is my gene to the module, right? It measures how co-expressed is my gene with this module eigengene. and therefore the resulting measure is a natural measure of module membership, and we denote it as KME, which stands for module eigengene-based connectivity measure. Remember, K denotes the connectivity, and KME is another measure of connectivity based on the module eigengene. So just to illustrate a nice application of this, here I use data from Päivi Päiväkanta's lab and Chris Plaisir, who studied familial combined hyperlipidemia in human subjects. And they had 75 adipose tissue samples from human subjects, and they had different clinical traits. There's disease status, there's total cholesterol, triglyceride levels, ApoB, and also they had identified in a linkage analysis one SNP that was related to hyperlipidemia status. So these are these clinical traits and SNPs, but the X, of course, the rows correspond to the module eigengenes that they found. So they found 28 modules, and now what they could do is they could correlate each of these modules with these lipidemia traits and the SNPs. And what do you see? There's one module, this TAN module in the middle, that contained 504 genes, and that module was actually highly correlated with disease status, correlation 0.33, also correlated with total cholesterol, highly correlated with triglyceride levels, and so on. Incidentally, in brackets, you always see the p-values. Now, let's think of it like an author. So let's say we submit now this paper and say we have found a module that relates to triglyceride levels in adipose tissue, and it also relates highly to this SNP. In principle, it's a very significant and exciting module. But then look at the p-values, 10 to the minus 4, and so on. If you have the mindset of a gene-based analysis where you correct for 50,000 comparisons, these p-values are not great at all, you know. However, look at the correlation coefficients. These are phenomenally large correlations. For complex traits having a correlation 0.4, that's pretty impressive. Even look at the SNP. I mean, a correlation between a SNP and a module eigengene of 0.33, that's a very high correlation. So what's going on? Why are the p-values so unimpressive? Well, the reason is because the sample size was low. These were only 70 human subjects, you know. And but why could my colleagues still publish it? Well, the reason is because they could argue that they carried out relatively few comparisons. There were only 28 modules, and these traits were highly related. So anyway you count it, even at a Bonferroni correction, which is the most stringent multiple comparison correction method, even when you applied the Bonferroni correction, these results were still significant, you know. And that highlights a major advantage that I mentioned earlier, that these module eigengene-based analyses avoid the multiple comparison problem. Yeah. There's a question, yeah.
In this correlation metric, is it that those values indicate the average correlation between the triglyceride value with those 504 genes? Like,
oh, yeah, just to be, very good question. The question is how do I define these correlations? And so the answer is as follows. We summarized each module by its eigengene, and then we take that eigengene and correlate it with that variable. It's literally a pairwise correlation coefficient. And later today tomorrow I will show you our code for literally producing that output, and so you can apply it to your own data. You can optionally report a bi-weight mid-correlation if you want a more robust measure, you know. But here I think these are Pearson correlations, and as I said, p-values. And the other thing that Peter Langfelder implemented here is that there's a coloring scheme which is very nice, right? Notice red indicates positive correlations, you know, and green would indicate negative correlations. And so it's actually a very powerful way to summarize your entire data set: SNPs, physiologic traits, and expression data. Yes?
And when you're calculating the correlation with a SNP,
Yes?
it's just measured as 0, 1, 2?
Yes, yes, exactly. So, many of you are geneticists, and when you have a genotype, there are different ways to code it. One way is to use what they call additive coding, where you count the number of the minor allele, for example, and then, yes, you have a trivariate variable, 0, 1, 2, and you just correlate it. And that's usually what we do. But if you want to use a recessive coding or any other coding, you can do that. The key is numeric coding. Yes?
What is some size?
Yeah, here the sample size was 70 samples, yeah. So it's low, you know. All right, moving along. As I mentioned, we can also relate module eigengenes with each other. So here I show pairwise plots between module eigengenes. And the only thing I want to convey with this figure is to say that module eigengenes can have high correlations with each other. You could have two distinct modules, the brown and, let's say, the brown module and the green module. And here actually every dot is a sample, and you see there's a negative correlation of actually minus 0.42. So modules can be related, which is a biologically reasonable thing, you know, because let's say modules correspond to cell types. Why would, let's say in brain tissue, why would oligodendrocytes be uncorrelated with the amount of neurons or so, you know? I mean, so modules can clearly be related to each other.
Steve?
Yes?
Can you back up one, please? What would be your interpretation of a high correlation to gray? If gray is the unconnected transcripts, but it's correlated to blue,
Yes.
what would that tell you about blue?
Yes. So just to be, so that everyone can follow it. So Mark just mentioned, see, here's the blue module and here's the gray module. The gray is what we call an improper module. Remember, it's a set of background genes. And when you look then at this pairwise plot, you actually see a strong relationship, and that is captured by the corresponding correlation coefficient, 0.78. So there's actually a high correlation between the gray and the blue. So there's two ways to interpret that. It could be that the blue module contains a lot of background genes, right? It's too large. Or it could be that the gray module contains actually a lot of blue module genes. Maybe the branch cutting wasn't good enough, and we should have included more genes into the blue module, you know. At this point, we can't answer that question, but one could look at gene ontology, you know, and let's say one of these modules corresponds to macrophages comprised of tens of thousands of genes that are overexpressed in macrophages. Then you could say, well, I should have included more, you know. It's a biologic question. But here's the good news, and I usually forget to mention this, but this is the one time I remember it, thank God. The critical thing about WGCNA is to get module eigengenes. It's not to get the modules, the subtle distinction. It's all about getting module eigengenes right. And it turns out that the definition of module eigengenes is utterly robust. Whether you cut a branch at the tip or fairly high up, the definition of the module eigengene is barely affected. Try it out, you know. Why? Because the module eigengene is a weighted average. Whether you use 500 genes to calculate the weighted average, or 200 genes, or 1,000 genes, the weighted average barely changes, you know. And so that is the really good news. As long as you focus on module eigengenes, you have a very robust analysis. And that also addresses the comment we had earlier about stability of the hierarchical tree. As long as you have a strong enough signal so that there's a module represented by a distinct branch, any way you get that branch, that set of genes, you will end up with the module eigengene which is at the backbone of the entire analysis. Why? Because remember, we use the module eigengene to relate modules to the traits, meaning disease status. We use the module eigengene for defining the measure of module membership, the connectivity. So you will get the definition then of disease-related modules, and to find disease-related hub genes is highly robust. All right, let me mention, while we're at it, this great advantage of module eigengenes, because it allows us to define a so-called blockwise module detection approach. Just to give you a background, when you use hierarchical clustering, it's computationally intensive, and so often you hit a barrier where you can't cluster more than, 50,000 genes or so, or 20,000 genes. But we, of course, apply it, WGCNA, to data sets comprised of 500,000 variables using a laptop, And so how do we do it? Well, later in the course, you will learn an R function called blockwise modules, And how does that work? So let's say we have 500,000 variables. Then the function would split these 500,000 variables into blocks of 10,000 variables each. There would be 50 blocks. And then, and how does it define these blocks? It does it by using a variant of k-means clustering. Why? Because k-means clustering is computationally fast. And all right, so you have these blocks, and now in each block it applies hierarchical clustering, the usual dynamic branch cutting, topological overlap, all of that. And so what do you end up with? You get 50 cluster trees. And for each cluster trees, you get modules. Now, it's critical then to compare the modules from different blocks, right? Because it could be that the blue module in the first block is highly related to the green module in the second block. And how do we know whether they are related? Well, we correlate the eigengenes. If the eigengenes are highly correlated, we merge these two modules, And that's, again, to highlight the use of the module eigengene, So therefore, we can, that's really at the backbone of this blockwise module approach. And we have several functions called blockwise modules, blockwise consensus modules. This blockwise approach is implemented in various tools, And it works very well. How do we relate modules to external data? Well, for example, let's say you have disease status as a clinical trait. There are several ways to do it. One is to define what we call a gene significance measure. By the way, let me step back and say, remember, we have now our modules, let's say based on 500,000 variables, and all we know is module 1, and we know its color, we know nothing about it. So now we need to annotate the modules. And the number one tool of annotating a module is by using these gene significance measures. So a gene significance measure is really a number, and high number means that gene is significant. For example, if you have a clinical trait, could be disease status, could be HDL levels, you can correlate each gene with that clinical trait and take the absolute value of the correlation. Or if you have two groups, you can use a t-test statistic and define for each gene either the absolute value of that t-statistic, or you get a p-value and you take minus log of the p-value. All of these are then gene significance measures based on a trait. Imagine, though, you don't have a clinical trait, but for one reason or another, you are really interested in apoptosis genes. How would you incorporate that into your network? Well, you would define a gene significance measure for each gene that takes the value 1 if you have an apoptosis gene, and it takes the value 0 if the gene is not known to be related to apoptosis. So that's now your gene significance measure. It's a binary measure, If you have a SNP, you can define a gene significance measure, as we discussed earlier, by correlating the SNP with each gene, as long as you use a coding of the genotypes. Now, once you have a gene significance measure, you can define a module significance measure by simply averaging it, right? Let's say you have the brown module, and then you just ask, what's the average gene significance measure of all the brown module genes? And that average I call the module significance measure. Now, incidentally, if your gene significance measure was based on a clinical trait, for example, using this correlation or t-test, then the result of this weighted gene significance measure is often highly related to the correlation between the module eigengene and the trait, So I find these are all, it's very robust, Whatever gene significance measure you use, chances are it allows you to find the modules that are most related to that trait, Yes?
But still, when you're building the network of the modules, it's still kind of unsupervised.
Yes, it is.
Okay.
Yes.
You don't use like a disease sitters
Yes.
to guide.
Yes.
Okay.
Yeah, so that's a very good question. Let me repeat it. So here I of course focused on the use of tens of thousands of genes, and I didn't pre-select them based on disease relationship, Having said this, some people have used this approach for analyzing, let's say, the 1,000 most differentially expressed genes, And if you do that, then I call that supervised WGCNA. Whereas the truth is, in most applications, I try unsupervised. Having said this, I could show you beautiful papers where people applied it to pre-selected genes. So we typically pre-select genes if we want to, based on things like their variance across the data or mean value. You could say, I want to focus on high abundance transcripts, various ways, Also, what I use the opportunity to mention that we assume that you process the data in the right way. These are somehow you used all the right pre-processing steps so that you arrived at biologically meaningful data. All right. Let me turn. Yes.
So the p-values that you presented for the eigengenes.
Yes.
It seems like it's PCA regression, essentially.
I wouldn't say it's essentially the same. I would say the logic is similar. Yeah. So just to rephrase it, there are various tools for using a trait and coupling it with principal component analysis. There's some partial least squares analysis. There are various tools, and so the logic is the same. Yes. So we take the data and we turn them into components in a shape or form, and then relate them. But the approaches, of course, differ by how they do it. So some approaches, as you mentioned, partial least squares would define the components in a way so that they relate to the trait. Whereas here we first find the modules by completely disregarding the trait and only in a second step related to the trait. Yeah. Yes, another question. Yeah.
Continue this question.
Yes.
Just feel like this method is kind of assumption-free type of
Yes.
disease regression.
Yes. Yes. Well, I mean, the logic behind it is that we want to find the low-hanging fruits, and that's the first thing, and we want to publish it, And right, imagine you start with an RNA-seq data set. They're expensive. You have 50 RNA samples, And then you use a trait to find trait-related modules, and you send it to Nature. So what will the reviewer say? It's a very nice study, but can you now get a second data set to validate it, When you use WGCNA in an unsupervised fashion and you say, look, here's the cluster tree. I find this gigantic branch It's highly robustly defined. No matter how I do branch cutting, here it is. These are 1,000 genes. This relates very significantly to disease status, let's say autism disease status. Even if I use the Bonferroni correction, then what did you just do? You convinced the reviewer that there's a strong signal in the data. There's the low-hanging fruit. Then you say, oh, by the way, the module is highly enriched with synaptic axonal guidance genes. The gene ontology enrichment makes sense. Suddenly you have a biologically credible story, and so the reviewers are a little bit more forgiving, hopefully, Yes. Let me take here a break so that you get a chance to get a cookie and so. And should we take a 5-minute break, Jenny? Is it lunchtime? Are you serious? Oh yeah, 5. Okay, then just let me continue then 5 more minutes. Sorry, I tease you sometimes, All right, so let's say then we have this exciting module. But now we're at the step where we want to find the key drivers, the interesting genes in that module. And before I go there, let's come up with some analogies. So here I show a graph of a co-authorship network of different mathematicians, and there's an edge between two mathematicians if they co-authored a paper together. And so if I ask you, well, who was the most influential mathematician here, then hopefully you see there's this guy, Paul Erdős, who connects to everybody. And so that's a hub, highly connected. And so in this context, one would say connectivity is clearly a valuable statistic for finding important nodes. Here's another application or illustration from Dr. Barabasi, where we show flight travel in the US for a particular airline. And so two airports are connected if there's a flight connection. And so again, you see these two cities that are hub airports. And again, these are very important nodes in that network. And these types of examples naturally lead us then to think, well, maybe hub genes in these gene co-expression networks are also important. It's a compelling intuition. There's only one problem. It's wrong, So, so hub genes with respect to the whole network are typically utterly uninteresting. If you have 50,000 transcripts, you form a network. Among those, you find the hubs, look them up, and you will yawn, So, but it is really, but genes that have a high connectivity in interesting modules, they can be very interesting, Coming back to this example I just alluded to, let's say you analyzed a brain expression data set, and you found a module that relates to autism disease status. Well, then if you look at the hub genes within that module, these intramodular hubs, well, they can be utterly interesting, they may play an important biological role. And so there are several papers that show that, But I just wanted to mention that important point. It's critical to focus on intramodular hubs in many of these applications. So, but how do we identify hubs? Well, we need a measure of intramodular connectivity because hubs in a module have high intramodular connectivity. And so the number one measure is defined as the row sum or the sum of adjacencies with respect to the genes inside that module, right? Let's come to a social network analogy. Let's say we have a high school and there are different modules in the high school. There's the module of the science club. And so now your intramodular connectivity in the science club indicates how many friends do you have in that science club module. And that's calculated by the sum of adjacencies. Now, the advantages of that measure, the main advantage is that it's really defined for any network, not just co-expression network, any network specified by an adjacency matrix. Could be a protein-protein interaction network. But the disadvantage is that it strongly depends on the module size. If you are in the science club and there are only 10 people in the science club, then your maximum intramodular connectivity is 10. Whereas if you're in the soccer club with 200 people, you have more friends, you know. So therefore, we often prefer the so-called KME measure, the module eigengene-based connectivity measure that I introduced to you a while ago. So the KME is defined by correlating each gene with the module eigengene, for example, the blue module eigengene. And as a correlation, this measure is naturally scaled. It takes values between -1 and 1. And also importantly, it maintains the sign information, right? I know is a gene positively related or negatively related to the module. This measure can also be used for annotating genes with respect to multiple modules. Why? Because you can have a gene that has a high KME value to the blue module, but also to the brown module. In essence, the gene is part of both modules, right? Why? Because it has high KME in both modules. And so in statistics, there's a terminology called fuzzy clustering. So I often get this question from audience members. Is it the case that a gene can only be part of one module at a time? And that question really requires a complex answer. When you define modules as branches, then yes, clearly a gene can only land in one branch. However, we actually don't ever look at the color labels. We look at KME, right? And so the answer is, of course, a gene can have high KME with respect to multiple modules, you know. All right, one question you can have is, well, given that we have these two measures of intramodular connectivity, what is their relationship? And the good news is that these measures are highly related. Here I have a formula where I relate the intramodular connectivity divided by its maximum value to the absolute value of the KME measure. Again, as I mentioned, there's actually some very nice mathematical theory, and you can prove that theory. But never mind the math. Let me show here a plot where I plot on the y-axis intramodular connectivity scaled by its maximum versus the KME measure on the x-axis for this power of 1. And notice I find almost straight line relationships. These are r squares of 1. So what does it tell me? It tells me whether you use the Km, the intramodular connectivity based on the adjacency, or whether you use KME for finding hub genes, you really end up with the same, you know. And so in practice, we select then genes based on KME. And as an example, we would use the 500 genes with the highest KME values and use them as to study their co-enrichment and say, you know, you upload these 500 genes into David or Ease or Ingenuity to find what's known about them.
Why couldn't you use the top 500 that have the highest KN?
Same thing.
It would give you the same
It gives you the same thing. That's what I'm trying. So whether you use KN or KME, you get the same 500. You know, the KME has this advantage that you could ask, give me 20 genes that have a negative relationship with the module. Right? The
Even for sign?
No, yeah. So the KME contains the sign information. And so the KME allows us to find genes that have a positive relationship and genes that have a negative relationship. Whereas KN doesn't distinguish. It only gives you, if you use a signed network, it only gives you the genes that have a positive relationship, you know. Yes. So as I mentioned, we define then these intramodular hub genes, and you can define them using a single network, and you say, well, there's this autism module, and I look at the hubs inside that module, and that's an exciting analysis. But you can also ask a different question. If I have two networks, the human brain co-expression network and the chimpanzee co-expression network, you can ask, well, which genes are highly connected hubs in the human brain, but not in chimpanzees, and vice versa. And so that's differential network analysis. So maybe should I stop here for lunch? Is that now it's lunchtime, and so we resume at 1:30, right? Is that correct? Yeah, at 1:30.